Well, welcome back to this room for the last keynote. We hope you had a nice time at the parallel sessions. I'm very pleased to introduce our last keynote speaker, Isabel Sola, who will present the development of a next-generation RNA replicon vaccine against SARS-CoV-2. Isabel has been working at the coronavirus field for more than 25 years and is currently co-director of the Coronavirus Laboratory at the National Center of Biotechnology. She made relevant contributions in the development of the first CoV reverse genetic system to engineer CoV genomes. The understanding of the coronavirus transcription, mechanism, and virus host interactions involved in viral replications and virulence. Her present interest is the development of a new generation of CoV-based RNA replicons as vaccine candidates. Welcome, Isabel.
Thank you very much for the introduction, and thank you also for the invitation to present our work to all of you. Well, is this working? Yeah, okay. Coronaviruses infect vertebrate animals. Birds and also mammals. In animals, they mainly cause respiratory and enteric diseases, while in humans, they are mainly associated with respiratory infections. Common ancestors of different coronavirus genera have been found in bats flying all over the continents. From bats, coronaviruses cross the species barrier to intermediate hosts, and then to humans, causing zoonotic infections that may become a pandemic. The presence of coronaviruses in animal reservoirs distributed worldwide makes new pandemics a realistic possibility. At this moment, 7 human coronaviruses are known. The 4 in yellow cause mild respiratory infections. Some of them, like 229E and OC43, are known 6 cystis, and they cause the common colds. The other 3 coronaviruses in red cause severe respiratory infections in humans, pneumonias that may be deadly, and all of them have emerged in this 21st century. The first, SARS-2, emerged in China in 2002 from civet cats as an intermediate host, and it caused an epidemic with 8,000 infections and 800 deaths. Later, in 2012, MERS coronavirus emerged in Middle East and is still causing sporadic infections from dromedary camels to humans, with a mortality of around 30%. At the moment, this MERS coronavirus doesn't transmit well from human to human, but viruses may evolve, and this virus should be subject to surveillance because the risk of being the new pandemic. And finally, in 2019, as all of us know, SARS coronavirus 2 emerged in China from an unknown intermediate host, and at this moment, more than 5 million deaths have been caused by this virus. Coronaviruses are single-stranded RNA viruses with one of the largest known RNA genomes of around 30,000 nucleotides. The viral RNA genome associated with the nucleoprotein is protected by an envelope that includes several structural proteins: the membrane protein, the envelope protein, and the most prominent, the spike or S protein. S protein is the responsible for binding to the cellular receptor to mediate entry into the cell, and is also the main inducer of neutralizing antibodies. That's the reason why S gene or S protein is the only component of most of the current vaccine that we are using. Different vaccine strategies have been demonstrated to be safe and to be effective against SARS-CoV-2 infections. Inactivated viruses use traditional techniques. They have limitations in terms of efficacy and may also have problems, safety problems, if inactivation is not complete. Using biotechnology, new vaccines have been engineered using subunit proteins or nucleic acids. These recombinant proteins are simple and easy to produce, but they require the combination with adjuvants to improve efficacy. Viral vectors have been engineered to express the S protein from the viral DNA genome. These vaccines have some advantages like the low production cost, and they are also easy to produce and transport. And the most innovative technology used for vaccines are mRNA vaccines. These contain a synthetic mRNA molecule coding for the S protein of SARS-CoV-2, which includes the minimal information to produce the antigen within the cells. This RNA is protected and is delivered into the cells by lipid nanoparticles. Although these vaccines have demonstrated to be safe and highly effective in the real world, they have some limitations, like the high production cost and the requirement of high cold temperatures for storage and transportation. The correlates of protection of SARS-CoV-2 infection are not well known, but there is information about the protective immune responses in convalescent patients. It was shown in almost all the patients the presence of neutralizing IgG and IgA antibodies against the S protein, along with a T-cell response against this S protein. But it was also found a T-cell response composed of CD4 and CD8 T cells against other components of the virus, the M, the N protein, and non-structural proteins, indicating that for protective responses, not only the S protein, but also other viral components might be required. RNA vaccines have a number of advantages. First, safety. mRNA cannot be integrated into the genomic DNA of cells. Then there is no risk for mutagenesis. mRNA is degraded into the cells by cellular RNases. So its half-life in vivo is short, and this reduces the risk of toxicity for the overexpression of the foreign protein. They are composed of a few components chemically defined, which simplifies the regulatory process. Its efficacy in terms of stability and translation is usually optimized by introducing regulatory elements and by chemical modification. And also, the efficacy of delivery requires carrier molecules. The large-scale production is feasible, but more economical options would be welcome. In addition to the current non-replicating synthetic mRNA vaccines that are being used in the pandemic, virus-derived cell-replicating RNAs represent a next generation of promising vaccine candidates. They have been optimized during viral evolution in terms of replication, gene expression, and also the evasion from the innate immune response. Today I will present our work in the development of self-replicating RNAs as vaccine candidates against coronaviruses. In our lab, we have been working for more than 35 years in coronaviruses, and we are interested in the molecular basis of coronavirus virulence by identifying virus virulence factors. Deletion of these genes from the viral genome leads to attenuated phenotypes, which are vaccine candidates, and also the identification of cell pathways involved in pathogenesis provides information about therapeutic targets for antivirals. Working with highly pathogenic coronaviruses requires biosafety level 3 laboratory and protocols like this one shown in the picture. This is the lab where we usually work at the National Center of Biotechnology in Madrid. To study viral pathogenesis and to develop vaccines, we have developed two essential tools. One infectious cDNA clone. This is a full-length DNA copy of the viral RNA genome that can be modified in the lab using molecular biology techniques. Our lab was the first one in the world in the year 2000 to develop a reverse genetic system for coronaviruses, which was challenging at that time because of the large size of the viral genome. And also small animal models that reproduce the severe respiratory disease and the death observed in human infections. These animal models are critical to evaluate the protection by vaccines and antivirals. Recombinant viruses are rescued in the lab from the infectious cDNA, which is assembled by joining synthetic DNA fragments. This full-length cDNA clone is introduced into bacterial artificial chromosomes and is transfected into mammalian cells. The RNA polymerase of the cells transcribes this DNA into an RNA identical to the viral genome. Then the virus life cycle starts and produces infectious viruses from the initial DNA. Genetically modified humanized mouse models have also been established in collaboration with other labs that express the human receptor of the virus. These mice reproduce infection of the lung, clinical signs, the inflammatory pathology, and finally the death of the animals. In the last few years, we have been working in the development of MERS coronavirus RNA replicons as vaccine candidates, which are replication competent and propagation defective. And these results are the proof of principle of vaccine candidates against SARS-CoV-2. Using the reverse genetic system, we generated a deletion mutant of MERS coronavirus without the envelope protein. This deletion mutant was propagation deficient, which means that the virus is lost along passages in cell cultures to titers below the detection limit. This is in contrast to the continuous growth of the wild-type virus. When the missing E protein is provided in trans by packaging cell lines, the growth of the virus is transiently recovered. This mutant is replication competent because it produces in the red columns genomic RNA and subgenomic viral RNAs to similar levels as the wild-type virus. These RNA replicons are produced in packaging cell lines in which The DNA coding for the replicon is introduced by transfection, and the packaging cell line is providing in trans the missing E protein. These self-replicating RNAs amplify themselves and produce other viral proteins to form these virus-like particles that deliver the RNA replicon into the target cells. But they don't have by themselves the E protein, so these virus-like particles are propagation deficient. Inside the cells, the RNA replicons self-amplify and produce high levels of viral antigens and form these virus-like particles that are similar in structure and also antigenically to the MERS wild type. Then, as antigens, they behave as the wild type virus and induce an improved immune response because they contain not only the S protein but also other viral proteins. Since these RNA replicons self-replicate, the initial dose that we need to immunize is lower, and that reduces the cost of vaccine production. And importantly, in the target cells of vaccinated individuals, there is no production of E protein. Then virus-like particles are propagation deficient, and this vaccine candidate is very safe because it cannot propagate from cell to cell. To increase the safety of these RNA replicons, other genes called accessory genes have been deleted from the genome. These genes are not essential for viral replication in cell culture, but they are involved in virus virulence in vivo because they interfere with the innate immune response. Then when we delete these genes, the innate immune response is increased and also the adaptive immune response that is finally protective. To illustrate the contribution of these accessory genes to virulence, a mutant without one of these accessory genes, without 4B gene, was evaluated in humanized mice and the clinical signs, the weight loss was studied, and also the survival of the animals. The red line shows this deletion mutant that initially causes a weight loss followed by a recovery, in contrast to the wild-type virus that causes a severe weight loss, and this is associated with the death of all the animals. In contrast with the attenuated virus, Delta 4B, all the animals survive, indicating that this gene is a virulence factor of the virus. And this attenuation is associated with a reduced innate immune response in the lungs of the animals, as shown in the red columns corresponding to the Delta 4B virus. The levels of inflammatory cytokines like IL-6 and chemokines like CCL2 or CXCL10 are reduced as compared to the wild-type levels, although both viruses replicate to similar extent. In addition to this recombinant virus without the E protein, essential for propagation, and without the accessory genes involved in virulence, we have also generated a replicon in which both sets of genes were deleted. This replicon, lacking 5 genes, is self-replicating because it maintains the replication machinery, which increases the antigenic dose and also the immunization potency. And it expresses several viral antigens, not only the S protein, but also M, N, and non-structural proteins to improve the immune response. These replicons lacking either 1 gene, 4 genes or lacking the 5 genes were evaluated to confirm the attenuation in vivo in humanized mice. All of them, in the colored lines, did not cause any weight loss, and all the animals survived, in contrast to the severe weight loss caused by the wild-type virus that also led to the death of all the animals. The replication competence of these RNA replicons lacking 5 genes was studied in animals, and it was confirmed in the orange columns that viral genomic and subgenomic RNAs were produced in the lungs. And as expected, this virus lacking the 5 gene was propagation deficient, also in vivo, because as shown by the orange columns, no infectious virus from this replicon was detected in the lungs of mice, in contrast to the high levels, millions of infectious viruses produced from the wild-type virus. And more importantly, the protection induced by these RNA replicons was evaluated in humanized mice by intranasal immunization with a single dose of the RNA, followed by a challenge with a lethal dose of the virus. All the immunized animals survived to the challenge with the lethal virus, and none of them showed weight loss, indicating 100% protection of the animals. In contrast, non-immunized animals weight lost, and all of them died after the challenge. And interestingly, this protection was unsterilizing immunity, because in the lungs of mice immunized with the replicon, no infectious virus was detected, in contrast to the high titers of virus obtained in the lungs of non-immunized mice. Then, immunization with the RNA replicons not only protected from disease and from the death, but also from infection and transmission of the virus. And this sterilizing immunity is mainly associated with the induction of a mucosal immunity by the intranasal administration of the replicon. Using a similar strategy, we are now working in the generation of a vaccine candidate, an RNA replicon vaccine candidate against SARS-CoV-2. We generated an infectious cDNA clone of this new virus and a collection of deletion mutants. And we have engineered a replication-competent, propagation-deficient RNA replicon by deleting a combination of genes required for propagation and for virulence. And 2 formulations are being assayed for the administration of these SARS-CoV-2 RNA replicons. One of these versions is virus-like particles generated in packaging cell lines that provide in trans the missing proteins required for replication. And in these packaging cell lines, DNA coding for the replicon is transfected, and finally, VLPs are recovered that deliver the RNA replicon into the target cells of vaccinated individuals. Another strategy is a cell-free synthetic formulation obtained by combining RNA transcribed in vitro with polymers to form nanoparticles. And we are working in the delivery of these RNA replicons not only intramuscularly, which is the preferred route by the medicament agencies, because that's the usual way of administration. But also we are convinced in the benefits of intranasal immunization to provide mucosal immunity and and then to provide sterilizing immunity, which is one of the problems of current vaccines. All of them protect from disease, protect from the death, but none of them protect from infection and also transmission. And finally, the acknowledgement to all the people in the lab who has been working in the past and also now in our vaccine candidates against coronaviruses. Of course, Luis and Juanes, who started the lab more than 35 years ago. And thank you very much for your attention.
Thank you very much, Isabel. That was fantastic. Well, if anybody has any question, please raise your hand. Here.
Thank you for your very interesting presentation. The research infrastructure, that they kept up the lab even when the urgency wasn't there. And in Holland, we reduced a lot of the investments in your type of research before the pandemic. And now, of course, we're sorry about that. But I have a question about, it seems like you have a very promising new way of developing these vaccine strategy. Do you collaborate with the pharmaceutical industry in order to prepare for future production?
Yeah, that's an excellent question, because we are a research lab and we have the capacity to innovate, but we need, of course, the collaboration with pharmaceutical companies. And we are now starting a collaboration with one pharmaceutical company in order to develop this next generation vaccine candidates, because we are aware that we need their help to go finally to the market, because we can generate a promising candidate and publish in a very good journal, but if you want a candidate to be in the market and to get to the arms of people, you need to collaborate with companies, and we are on that way.
Well, thank you very much for your participation, and thank you, Isabel, for your contribution.
Thank you. It's a pleasure.
And now it's time for the closing remarks. So I would like to welcome again the PEROSH Chair, Pauline Bongers.
Well, I think it has been a quite intensive day for all of us, and I was very, very pleased with the high level of contributions. In particularly, I would like to mention also the high level of contributions from the keynote speakers. I think we just learned a lot from this explanation about the vaccine strategies, and understand a bit more what's going on with this large promise of developing a vaccine which is also protecting against infection and transmission. And also this morning I thought it was very interesting to have a very nice presentation on one of the core subjects of what is interesting to us, and also the nice and very emotional, committed story from inside about the shortage of masks and our role as ARCH institutes to really provide support in such a pandemic. I'm very pleased that if I can look at you, most of you really didn't withstand the how do you call it, the attraction of the sun outside, because for us out north this is really an attractive alternative option to really catch a little bit more sun before going into autumn and winter. But I think everyone really stayed and listened and participated, so very well, thank you. I would also, maybe at home people are still looking at us, following the live stream. I actually had a short view and it really looked very professional. You could very well see the presentations and the presenters in this room, so everyone who really was with us online also had the information from the keynote speakers. And I think our next research conference will have a tough task to really surpass the positive gender balance in this in this keynote presentation lineup. So I think we had a very pleasant birthday party, and of course the birthday party was extremely well organized, and I really can't say enough to Mercedes, Marta, and all your colleagues from this ENSST who made this all possible. And so a very big applause for them. Jan Michiel also did quite a bit, I have to admit. So he was a big support, because he's the continuing factor who can transfer all the experience and knowledge from one conference to the others. But yeah, I think we are all very pleased that it was so successful, our first step on this new professional normal. So yes, I think we can look back with a lot of optimism. Then before you can actually go to the sun, I would like to invite Nader and to have a word. Maybe you can come too as a colleague, to have a word to where the next conference will be. So then I will leave the podium and leave the word for Nader, and I think after that we give the word back to the chair to really say the last words. So, please.
Well, thank you, Pauline. And as Pauline said, now Sweden is ready to take over the Olympic torch from Spain and run towards 2023. I assure you that it's not an easy task, especially after attending this well-organized and highly interesting research conference offered to us by Spain. Thank you, Spain. It was very interesting to all of us. But I promise you that you are going to have a very nice and a very high qualitative research conference in Sweden. We have 2 years to prepare it for you. And I assure you that you are going to enjoy the beautiful nature of our capital city, Stockholm, which is often called the Venice of the North, with more than 20,000 islands in the Stockholm archipelago. So welcome to Stockholm, you and your colleagues and your institutions, and we are very eager to see you in 2023. Thank you.
Thank you. Thank you very much. So we have come to the end. I would like to give congratulations to the organizers of this great conference and to all the people that has been working behind the scenes, because this was perfect. I would like to thank you, all the speakers, for their great contributions. They were brilliant. And of course, I would like to thank you all for participating. I hope you had a nice time in this conference. And despite all the inconvenience caused by COVID-19. and we wish you a safe travel home. For those who are staying in Madrid, we hope you enjoy the city. And we also have organized a farewell coffee at the same place where you have lunch. So thank you all and goodbye.